right, so good afternoon, and thank you for attending the second seminar in the Iowa Nutrient Research Center's Spring Seminar Series, Focus on Phosphorus. I am Kay Stefanik, Assistant Director of the Iowa Nutrient Research Center. Today, our speaker is Dr. Barbara Cade-Mennen with the Swift Current Research and Development Center in Saskatchewan, Canada. Probably one of the nice things about having these virtual meetings, we can bring in people from all over US, North America, potentially even the world. Before I get into introducing Dr. Cade-Mennen, I have a few items that we should go over first. So if you missed the first seminar by Dr. Justin Chaffin, there is a recording posted through the INRC website. We also have two remaining speakers in the series, Dr. Antonio Mallarino with Iowa State University. He'll be presenting on March 23rd. And then Dr. Chad Penn with USDA ARS and Purdue University, who will be presenting in April. This seminar is going to be recorded and posted to the Iowa Nutrient Research Center's website, along with past seminar recordings. As we go through the seminar today, if you have questions for Dr. Cade-Mennen during her talk, please type those into the chat box and I will relay them to her at the end of the seminar. So getting on to introductions. We have Dr. Barbara Cade-Mennen with us. She is a research scientist with Agriculture and Agri-Food Canada, Swift Current Research and Development Center in Swift Current, Saskatchewan, Canada. Dr. Cade-Mennen earned her PhD in soil science from the University of British Columbia. Her research focus is on nutrient cycling and minimizing nutrient loss from agriculture, with research projects such as looking at the effects of management practices on soil phosphorus forms and cycling, soil acidification from long-term nitrogen fertilization, practices that minimize nutrient loss from land to water, and phosphorus forms and cycling in aquatic ecosystems. Dr. Cade-Mennen's talk will be on phosphorus forms and cycling in agricultural soils of southern Saskatchewan. So with that, I will turn things over to you.
Thank you. Well, welcome to my talk and thank you for the invitation. And as greetings from chilly Saskatchewan, where with the wind chill, it's -30, which is about the same, of course, in Celsius and Fahrenheit. So as I said, I'm going to talk about phosphorus. And is my, of course, things, oh, there we go. So why do we study phosphorus? Well, of course, it's an essential nutrient for all organisms. As the slide from the International Rice Research Institute shows, plants without phosphorus don't develop very well, and you start to see signs of phosphorus deficiency. Of course, the other reason we study phosphorus is because too much phosphorus causes water quality problems. And this is a nice photo from David Schindler's lake study experiment where they put a barrier in and added nitrogen and carbon to one side and carbon, nitrogen, and phosphorus and showed it triggered algal blooms. Of course, none of us would ever get permission to do this nowadays in a whole lake city system. But anyway, these are the reasons why phosphorus is interesting. So my talk is going to give some background information about phosphorus forms and cycling, phosphorus pools and methods, then talk about some of my research results in soils and runoff, and then a summary. And I have discovered over the years that I probably look at phosphorus a little differently from most people. So you're going to get to see my vision of what phosphorus cycling looks like. So I'm going to start with the background about phosphorus forms. Let's start with orthophosphate or phosphate, and this is H2PO4, HPO4, etc. Single phosphate group with 4 oxygens. We also, but it's not the only organic or inorganic phosphorus form, although it is the most bioavailable. We can also have polyphosphates and pyrophosphates. So these are chains of orthophosphate linked by anhydrous bonds. They can be, pyrophosphate is 2 phosphates, polyphosphate is 3 or more. One thing you'll note, though, that I haven't mentioned is P2O5, because this compound does not exist. We need to stop using it. It's a very old term dating back to the 1800s, and I don't know why it's still in existence from a chemistry point of view. Anyway, that's my little beef about that. So we talk one thing about phosphate, and actually some of the compounds linked to phosphate as well. They lose they're deprotonated at pH 2, pH 7, and pH 12.7. So at the pH of most soils, we're going to have some combination of H2PO4 with a single charge and HPO4 with two charges. And of course, below pH 5, phosphate tends to be associated with aluminum, iron, and manganese, above pH 8, calcium and magnesium, and in between, it's some combination of all of those. Common phosphate minerals range from including aluminum and iron phosphates, lots and lots of calcium phosphates of various solubilities, with hydroxyapatite being the least soluble, and monocalcium phosphate and brushite probably the most soluble. Struvite, which is something that's had a lot of interest from industry as it's being made from wastewater treatment plants. And of course, my personal favorite, which is turquoise, which is a copper aluminum phosphate. Moving into the organic forms of phosphorus, this is phosphate linked in some way to an organic group. These can be divided based on the way they are linked. So monoesters, as the name implies, have one ester bond, which is what would be at the corner here, for every phosphate group, and they're linked through oxygen. This is phytate, which is fairly well known for studies because it is the most common grain storage compound, and ends up in manure, and of course with all these charges binds to soil. But there are lots of other monoesters, including sugar phosphates, and these are involved in storage and signaling. We also have the diesters. These have two ester linkages from phosphorus, and include the molecules of life, such as RNA, DNA, and phospholipids. And the third major category of organic phosphorus compounds are phosphates. These have direct C2P bonds without oxygen, and they include both natural and man-made or synthetic compounds. Glyphosate is a case in point with no oxygen there. Phosphomycin is another one that's interesting. It's actually an antibiotic produced by Streptomyces, and it was one of the suite of antibiotics Selman Waxman won his Nobel Prize for in 1952. It was used to treat tuberculosis. So why do organic P forms matter? Well, because the form is going to affect phosphorus cycling and plant availability. They do not all behave the same in the soil. For example, if we have phytate with all its charges is very different from phosphate with two charges here, or with only one, depending on the pH, versus polyphosphate. These are also, so having these different charges means they're going to bind to the soil, in the soil differently. They're also going to require different enzymes to cleave phosphate from pyrophosphate versus phytate. We get to soil cycling. We have biological processes, immobilization and mineralization, so that's organic phosphorus to phosphate. Chemical processes involve inorganic and organic compounds, and these include precipitation and solubilization, and adsorption and desorption, with precipitation occurring in solution and adsorption occurring on mineral surfaces. And it's an S note. These are often studied only for phosphate, but most organic phosphorus compounds will also be affected by precipitation and adsorption. This leads us to this example of soil phosphorus transformations. So imagine, if you will, that this is a soil particle here and a soil particle here with a root or hypha and a microbe. So tiny, tiny scale. And within, we have the soil solution in the center and all the processes that I mentioned. We can have absorption and desorption. We can have uptake or release. We can have mineralization, absorption and desorption of organic and inorganic compounds. And what makes phosphorus so difficult to study is all of these processes can occur simultaneously. As a phosphate is released, it's not just going to hang around in the soil solution. It's going to do something. It'll precipitate or absorb or be taken up, which means that a lot of these reactions occur so quickly, it's hard to measure them. So let's talk about phosphorus cycling and phosphorus pools. Phosphorus transport is diffusion limited, so it depends on temperature, moisture, and concentration. And as phosphate is taken up from the soil solution, phosphate will move into the soil solution to replace it. And we talk about labile P as what is in the soil solution, including phosphate and organic P moves by diffusion, and it's easily converted to phosphate for uptake by plants or microbes. Moderately bound phosphorus is more tightly bound. It's usually precipitated or adsorbed, again, both inorganic and organic forms, slower to move into soil solution and not readily plant available. And then we have tightly bound, very difficult, again, both organic and inorganic forms. And most of what we look at, especially if it's soil test P, is simply labile P in soil solution, and particularly for phosphate. Looking at phosphate, there's more soil P cycling, phosphorus pools. We can measure total phosphorus, which is obviously all the P in a sample, usually measured by some sort of digestion method. We can have total organic P, which is all the P associated with carbon or organic matter. We measure it by processes involving ignition to release all the phosphate from organic phosphorus and some kind of extraction. And then we have various soil test P, which is probably what most people are more familiar with, and that includes Olsen P, Bray P, Malick P, Cologne P, all sorts of different methods developed in different regions. What's important to note for soil test P is it doesn't measure plant available P. I often, when I'm reviewing, I will see people talk about having measured available P, and we don't actually have a test for available P. Soil test P needs to be calibrated with field trials, and it really is a response to fertilizer rather than truly what is available to crops. The other thing to note is soil test P is a very small portion of total P. So for example, and this is typical of the soils I study here in Saskatchewan, total phosphorus can be 1,000 milligrams per kilogram, of which half is organic phosphorus, and soil test P is maybe 30 milligrams per kilogram, so 3%. And it's important to note when we look at that the soil test phosphorus that most people measure and is one of the main forms people look at is just a tiny pool. It's here at the absorption and desorption. We need a lot more information about what isn't measured by soil test P, because that 97% has to be as meaningful as that 3%. Other important factors for soil phosphorus cycling. Well, soil pH, as I mentioned earlier, we're going to have affected by, depending on the pH, iron, aluminum, and calcium come into play, and magnesium, of course, down here. And the maximum solubility of phosphate is around pH 6.5. So shifts in soil pH become really important for controlling phosphorus or releasing it. The other factor that's important is soil depth. Phosphorus is always stratified. Even with tillage, it will be stratified just where it's tilled, or mixed maybe just where it's tilled and stratified below. So looking and comparing soil depths doesn't necessarily mean anything, but it becomes really important as we measure for concentrations, especially for soil test P. So these are some examples that I'm actually going to be discussing a bit later from a study, but annual cropland versus tame pasture and native prairie pasture. And you can see that Olsen P is much, much higher in the top 10 centimeters than it would be below. So sampling from 0 to 10 centimeters is going to give you a very different result from sampling from 0 to 30 centimeters, because it's going to dilute phosphorus concentrations, and it will always come back that you need to add more fertilizer if you sample deeper depths. And that becomes a problem if you're putting too much phosphorus fertilizer on. Other factors with biological cycling are that we have plants and microbial P strategies for getting phosphorus. One is that there's different rooting systems to exploit different soil volumes, and this becomes important with choosing crops if you want to maximize phosphorus or taking phosphorus out of the soil. We can also, plants and microbes will also alter phosphorus chemistry, converting phosphorus forms by either producing organic acids, which can then solubilize, absorb both inorganic and organic phosphate, changing the rhizosphere chemistry. They also produce enzymes, phosphatases to mineralize organic P. And as noted, these are specific to forms. For example, phytase for phytate. It is not going to mineralize another form. They can also make friends, rhizosphere bacteria, particularly P solubilizing bacteria, and those often work because they produce organic acids. Also mycorrhizal fungi to explore different soil volumes. So how do we study soil phosphorus? A method will depend on what we want to characterize, of course. So do we want to know general pools? As I said, relatively soluble, labile, insoluble. Do we want to predict fertilizer response or environmental risk? Or do we want to know the specific form? Does it matter to us whether it's phytate or apatite, or do we just want to know salt SP? Because all of this will determine methodology. If we just want to look at general chemistry, those are generally with some kind of extract that can be precise or general, followed by either colorimetric analysis or ICP analysis. ICP will measure total phosphorus in solution in whatever extract. Colorimetric will measure molybdate, reactive, or whatever your color reagent reactive phosphate. But if we want to know more than just general pools, we start to go into advanced techniques. This can include NMR spectroscopy or some kind of XANES, so X-ray absorption near-edge structure spectroscopy. And this is the NMR facility I use at the University of Saskatchewan. And this is the Canadian light source. So this is synchrotron based and very high powered light source for things. So techniques to characterize phosphorus forms. I'll get into more detail about XANES. This is where an absorption of an X-ray photon by core level of an atom causes emission of a photoelectron, which emits at, and you measure that emitted energy. We can do this at the K edge or L edge. It is direct, non-destructive, requires very small samples that are finely ground. However, it's synchrotron based, and while access to synchrotrons has been improving steadily every year for the last 15 years, they're still not everywhere. I mean, there's one in Canada, there's one at Stanford, and one at Brookhaven, and a few others at the US. The one at Brazil is becoming known for phosphorus, but they're there, but they're not something everyone is going to use. You can measure both organic and inorganic phosphorus, although it's a better technique for inorganic phosphates, and it works better with high P samples. So detection limits are really an issue. To do a typical XANES experiment, you start with the sample, you grind it, you mount it on a copper double-sided tape. This is a Canadian dime, which is the same size as a US dime, just to give you a sense of space. We then take it to the light source, put it into the beamline, analyze the sample, and it gives you a spectra, which you also analyze various standards, and then you do line fitting to figure out what kind of phosphorus forms you have. And as you see, the different, while the standards have lots of differences, the spectra themselves don't necessarily do. We can also, and this is done at sort of millimeter scale, but we can go even smaller than that and start looking at microXANES and look at hotspots. So we get a hotspot of one here. We can look at whether it has, look at calcium, iron, aluminum, silica, phosphate, and then put them all together to see what phosphorus is associated with. It doesn't necessarily tell you specific forms, so it doesn't tell you what kind of calcium phosphate, but it does tell you that phosphorus is associated with calcium. Another major technique is NMR. This is the one I probably use more than anything. Is you put the sample into a giant magnet. So your NMR facility, you have a magnet here, and basically the brains behind it here, the software, and then you sit at a computer and process it. And so you align the phosphorus nuclei into a magnetic field, you excite them by a radio frequency pulse, and as they relax, they release energy, which is what we measure. We can do both solution and solid state, but for most phosphorus compounds, solution is better. That's we extract it and put it into solution. For soils, this is also important because iron will cause broadening, and most soils have iron in them. There's only P31 as a nucleus, which means it's 100% abundant. And which means our NMR results are quantitative if we measure the area under peaks. Instruments are more available there. I don't think there's a university in North America that doesn't have an NMR, although whether we get time for the non-chemists because we need longer experiments. It's a typical experiment. Again, you take the sample, we extract it, put it into liquid, put it into an NMR, and just for scale, I'm 5'5" on a really good day, and I have to stand on my toes to put the sample in at the top of this magnet. We do a pulse acquisition delay experiment as many times as we want. That's called a scan. And that gives us a spectrum that looks like this. And this is a real sample of forest floor where you can see all those different compounds that I talked about. The phosphonates with a direct CP bond are down here with less shielding of the nucleus, and outfield where there's high shielding of the nucleus, we start seeing polyphosphates. We see phospholipids, DNA, pyrophosphate, etc. I said if we measure the area under these peaks, it's a quantitative method. All right, let's move into environmental samples a bit more. Some of the questions that are asked in research, and I've certainly done research for all these areas, are what are the forms and concentration of phosphorus in samples with different land use management or land use or in manures or in water? How can we manage phosphorus fertility and phosphorus pollution risks from runoff? And what factors control phosphorus cycling, including landscape, climate, soil age, aquatic environments, etc. And I'm going to talk about results from 3 of my research projects, one on legacy P and agricultural soils, one on land use and soil P, and one on phosphorus and snowmelt runoff. So just to let you know, I'm based here in Saskatchewan, which is in the center of Canada, approximately, well, I think probably the center of Canada is maybe closer to Manitoba, but it's the province that's easy to draw and hard to spell. But yeah, kind of a square, rectangle here. I'm based actually in southern Saskatchewan. So Haver, Montana is here, and it's about a 3-hour drive for us to there, just to give you a sense of where we are. Regina is our capital. Saskatoon is the largest city, and sort of north up here and north, that's boreal forest, but down here it's shortgrass prairie. The region I'm in, and Swift Current here, is it's dryland agriculture. There's almost no irrigated agriculture here, although there is more so if you move into Alberta. It's wheat-based with various, usually spring wheat, but there's also rotations involving the pulse crops we most commonly use are lentil and pea. Flax is common, canola, and mustard is a big crop around here. We don't grow corn or soybean here. It's too cold, but that's coming in more to Manitoba and Ontario. I'm in a semi-arid region here. We get 300 to 400 millimeters of precipitation, about a third as snow, but that's highly variable. And our soils are in the Canadian classification system, Orthic brown turnisems, or Abrida capilleboros at the USDA classification system. So legacy phosphorus can be defined as elevated soil P concentrations that result from prior land management. So this can be from applying fertilizer based on fertilizer recommendations. It's well established that not all the fertilizer that's applied every year is taken up by plants, so it gets retained in the soil. It can also apply from disposal of manures. And it is of concern because it builds up to really high proportions. It can contribute to phosphorus loss, even despite conservation measures. It can be, if it's not soluble, it can be moved by soil erosion, or it can, if it is soluble, it's just going to be lost in runoff. But the other reason it's of interest right now is trying to understand what makes this available to crops, because it could reduce the need for phosphorus fertilizers. And of course, phosphorus fertilizers, as we currently use them, are mined from rock, and that's a non-renewable resource. So trying to find ways to cut down on rock phosphates is always a good thing. Long-term studies that look at phosphorus buildup and drawdown are needed to understand changes in phosphorus forms. So I started here in Swift Current in 2008, and one of the big draws for me to come here were the old rotation plots. I inherited a great experiment related to phosphorus. These were initially established in 1967 with all sorts of, you can see the overview of all the different rotations together, and some of the ones that we paid attention to are here. Continuous wheat, lentil wheat, some with forage, some with fallow. They are all phases are present for anything that's, so of course continuous wheat, that doesn't matter, but if there's fallow wheat, all three of those phases will be present each year, and they have different combinations of with and without nitrogen fertilizer, and with and without phosphorus. So I'm going to focus on continuous wheat, both with N and P, and then without N and just with P. And nitrogen fertilizer is approximately 32 to 50 kilograms per hectare. It's based on spring soil tests every year. It was ammonium nitrate from 1967 to 2008 and has been urea since 2009. Phosphorus is around 10 kilograms per hectare. It's not based on soil tests. It's just a flat rate that was added every year. And it is monoammonium phosphate. And then in 1995, they stopped adding phosphorus on subplots in these major plots. So you can see it's about the top quarter of each. And this was not done out of an interest in phosphorus. In 1995, nobody cared much about phosphorus. This was done because cadmium was being added to these plots, and wheat is sensitive to cadmium. And cadmium is part of most phosphate fertilizers. However, they kind of stopped monitoring cadmium for whatever reason about 2005, but they kept the no phosphorus experiments going, which for me was really interesting. So this is for these continuous wheat plots, that's given us a combination of plus phosphorus and nitrogen, plus nitrogen, no phosphorus, minus nitrogen and phosphorus, minus nitrogen, no phosphorus. So I'm going to talk about two studies, one that was published in 2015 that used, the other thing I'll note is the samples were collected and archived. The soils were archived back to 1995. So when I started in 2008, I could go back and restudy all the different soils with the kind of chemistry that I do. So the first time we talk about samples from 1995 and compared them to samples from 2010, just with the plus nitrogen with and without phosphorus. And then I'm also going to talk about a study from 2016 where we included the plots without nitrogen. So in our 2015 study, we had, these are the 0 to a 7.5 centimeter depth. We sample at 2 depths. And so we have the blue is 1995 with all the fertilizers, and then red is 2010 with continued phosphate fertilizer, and then the green is without, where we stopped at 1995. And so you can see that both for OLCP and malic P at the surface, phosphorus concentration, soil test phosphorus concentrations have increased with added fertilizer and have decreased when you stop. But what's interesting is it hasn't made any difference to yield. There's no differences. And that was kind of interesting to us because clearly we're drawing it down. Is it just that what's still here is sufficiently available, or is there something else going on in these plots? So we looked at the samples from 1995 to 2010, and we looked at total phosphorus, which didn't change. And these are 2 depths now, 0 to 7.5 and 7.5 to 15. So total phosphorus really has not changed much between 1995 and 2010. Organic phosphorus has, though, and it increased mostly because inorganic phosphorus was decreasing. So the plants were definitely pulling out inorganic phosphorus, which was causing organic phosphorus as a percentage of total to increase. We also found, when we looked at the XANES analysis, some big changes in chemistry. So aluminum phosphates were much, much lower without phosphorus fertilizer, whereas iron phosphates were a lot higher. And we had a shift in 1995 with a lot of hydroxyapatite phosphorus, which is relatively insoluble calcium phosphate form. But by 2010, we were seeing more of tricalcium phosphate. This study was published in Environmental Science and Technology, 2015. We followed this up because we were still curious what was going on. So we collected samples in 2016 and saw the same thing. We're still not seeing on a year-by-year basis any significant change in yield, despite having no phosphorus fertilizer since 1995. We see some differences with nitrogen, but no differences with phosphorus. So one of the things we decided to look at was, of course, we've heard rumors about soil acidification coming out of Montana and other states. So we looked at cumulative ammonium addition. And one thing, of course, when we stop adding MAP, it does decrease the ammonium as well. And what we see is an interesting change, significant change in pH that amounts to half a pH unit per 1,000 kilograms of ammonia added. And this has changed the soil chemistry. Exchangeable aluminum and iron have both increased with decreasing pH, and the opposite is true for calcium and magnesium. And these are, of course, strongly correlated to pH. And this becomes really of concern for aluminum in particular due to iron or aluminum toxicity, which wheat is really sensitive to. When we start looking at phosphorus chemistry, total phosphorus, of course, echoes what we see with the fertilizers, and not much difference with nitrogen. With or with, it reflects phosphorus more than pH. Same with Olsen P and Malick P, but organic P is completely tied to pH, which was really interesting. At lower pH, but it makes sense in terms of absorption and precipitation. These forms will bind as pH decreases. We also saw changes with phosphorus forms of NMR and XANES, which I won't get into today. The other thing that was really important is it has changed the soil microbial community and changed enzyme activities, everything related to phosphorus cycling. So in summary, when we look at these long-term plots, continuous wheat in 1995 versus 2010, with just the nitrogen or with and without nitrogen, we saw no significant changes in yield or in grain or straw phosphorus from stopping P fertilizers on a year-to-year basis. Overall, we see some differences, but yield in these plots is more related to nitrogen and rainfall. But what this tells us is, and this is continuing even to recent years, that the plants are accessing this legacy P somehow. We see significant shifts in pH, altering phosphorus chemistry, microbiology. This pH changes mostly in the surface 10 centimeters. And this is really important because certainly most of our producers around here sample deeper. They sample to a foot when they submit their samples, and they might miss some of these changes. We also know these pH changes can be linked to plant diseases, and they've reported similar changes in Montana and other states. Also to note, the lentil wheat plots have not received only get the nitrogen when the wheat phase, so half as much nitrogen, and the pH has dropped just as much. So something to follow up on, and we are. Moving to land use now, and common land uses in southern Saskatchewan are native grassland, tame grassland, which is crested wheatgrass, and this was planted in the 1930s on land that was cleared for agriculture, but then of course was affected by drought and erosion. We also have annual cropland, and we have these roadside ditches, which are interesting. So the objective of this was to look at phosphorus and other things in these different land uses. What we had were 4 blocks that contained all 4 of these land uses within a very short distance from each other. We sampled to 0 to 30 centimeters depth with lots of different analyses. If we look at phosphorus with NMR, these are the different phosphorus forms. What we have is the blue here is annual cropland, the red is native grassland, the green is the tame pastures, and the purple are the roadsides. And we can see there's some pretty significant differences with land use for phosphorus forms. We also see some pretty significant differences in terms of enzyme activity with while inorganic phosphorus is high for both annual cropland, which is expected, they're fertilized, and but also high in roadsides, which wasn't expected. Enzyme activity is also much higher. And we also looked at the microbial community abundance and metagenomics for genes, which I won't get into completely here. But what we see is really interesting is annual cropland had the highest abundance for microbial communities, followed by roadsides, and the pastures, both the tame and native, were similar to each other and had the least. What this shows was that degree of disturbance was really, really important. And so the roadsides and annual cropland that are most disturbed seem to be most adaptable in terms of the microbial community, the genetic abundance, and even enzyme activities. Now I'm going to move into runoff studies. So again, we're in the Northern Great Plains and we're in what's called the prairie pothole region. So these are seasonal fill wetlands, many of which are drained for agriculture, but they don't have a connectivity except in runoff, spring runoff. And for us, winter is 4 to 5 months per year, and on the good years, and it's 6 to 7 months on the bad years. Annual precipitation, as I said, is about a third of it is snow. Most of the rainfall we get is patchy, as this slide shows really nicely, and as in summers it's lost to evapotranspiration, so and it's localized. So the main runoff events for us are as snow melt over frozen soils, and tend to be a very 1 week every spring, everything goes crazy. So a lot of the management practices that are developed to control runoff for summer storms, or in the US where it doesn't get quite as cold, are not necessarily effective on the Canadian prairies. And so what we did was some studies to look at what would work. So the objective of the study was to look at physical forms of phosphorus and snowmelt runoff with an event in April 2011. This was the major snowmelt runoff event for the year, and we compared 2 different treatments: annual cropland for wheat, which is a wheat canola rotation, and then we also had a pasture summer graze control. And in this case, he has his cattle out there. It's a beef operation, and they're out there for maybe 2 weeks a year in that particular pasture. And we compared it to 2 other pasture management things. One is called in-field winter bale grazing, where the producers actually put bales out on their fields in the winter time and graze the cattle out there. This has some benefits because there's no cost to clean up corrals and dispose of manure, and also because the animals are not clustered together, they tend to be healthier. But that does, and the thought for this is it's going to capture nutrients because they'll be directly deposited onto the ground. We contrasted that to the pastures, exactly the same pastures we use for the fertilized cropland control, but also to fall manure spreading. So our sampling was done in spring snowmelt. Here's my postdoc at the time out there sampling. So don't complain about field conditions if you're not standing in the field during snowmelt runoff. But we had automated samplers. We had flumes, and you can see the pasture control versus the bale grazing. Gives you a pretty interesting idea of the results we're seeing. These were all brought to the lab, filtered within 24 hours, and then were analyzed for all sorts of things. In case of phosphorus that I'll talk about now, we used ICP to measure total dissolved phosphorus, colorimetric for dissolved reactive phosphorus. We did digestion for particulates, and I did phosphorus NMR analysis on both the particulates and the dissolved samples. Let's look at physical forms. This is the dark green is the pasture, the lighter green is the cropland, and as you can see, there were some significant differences. This whole snowmelt event lasted for about 5 days. I've numbered the days rather than dates just because our pasture tended to start a day before our cropland, just because they were different. One was south facing and one was north facing, and so they had about the same length, but not exactly the same day. Some significant difference for treatment, and generally it decreased over time. Particulates, on the other hand, although if you note the scale, it's an order of magnitude lower, but pastures were always higher than croplands in terms of particulates, and a much lower form. Let's compare that to the bale grazing, or to the pasture treatments. So all of here, the first is the bale grazing, then we have the manure spreading, then we have control. And this control is what I showed in the previous slide. So as you can see, fall manure spreading significantly increases total dissolved phosphorus. whereas the bale grazing increases the particulate phosphorus. So order of magnitude higher. This is almost 4 times higher. Looking at the NMR, these with NMR, and what I did was I looked at samples from day 2 and day 4. So Pasture and Cropland on day 2, and then Pasture Cropland day 4 for orthophosphate, so phosphate, the inositol phosphates, including phytate, other monoesters, the phosphonates, total polyphosphates, and diesters. And there were some significant differences, although dissolved phosphorus is mostly orthophosphate, which decreased with time. In contrast, particulates had a lot of organic phosphorus. The pastures had more orthophosphate, but the croplands were mostly losing organic forms of phosphorus in their particulates. Focusing though in on the phosphonates from the croplands, there was something really interesting. We can see glyphosate moving off, and that's because it's often used in the fall to desiccate crops, and so it's standing on the stubble and it's just washing off in snowmelt runoff. Looking at the pasture treatments, this is the dissolved phosphorus from the first, so bale grazing, manure spreading, and the pasture controls. And see, fall manure spreading adds a lot more orthophosphate, but we do see dissolved organic phosphorus showing up in the diesters in particular for the bale grazing. But what's even more striking is that the diesters that start showing up with the bale grazing in the particulates, and the concentrations, flow concentrations, of course, this is becoming a significant portion now of what's going on. And looking at these further, we looked at fecal coliform counts. So the blue dots are our colony of fecal coliform counts from bale grazing and control, at which corresponds to the diesters are things like DNA and phospholipids, the biological phosphorus. And what we observed was that the bacteria, if you get a cow patty that's covered with snow, it's insulated and it stays alive and well. And those bacteria they cling to organic matter and just float downstream. Fortunately, we didn't see any incidences of disease downstream, but it was definitely there. Something else we did was look at some in-lab runoff, where we looked at different fertilizer applications, including broadcast, deep place, banded, using simulated snowmelt runoff. And under conventional tillage, unfortunately, the study was done in 2 different years and wasn't sort of coordinated very well, but we have controls versus seed placed and deep banded, a lot less dissolved reactive phosphorus than broadcast, clearly, much more. And then under the no tillage, we split this out a bit more and looked at both the inorganic phosphorus, so the DRP in the top, then the organic in particulate. And what you see again is the broadcast is definitely contributing more to runoff than anything that's put into the soil itself. And again, effects are going to change with tillage practice. So in summary, back to my slide of all these reactions, the point they call from all of this, phosphorus is an essential nutrient for all organisms, and if we're going to manage it properly, we need to understand it. And it's probably, compared to say nitrogen or carbon, there's still a lot of gaps in our understanding of the phosphorus cycling. Particularly related to biological phosphorus, we still spend way more time analyzing and looking at a very minor component of the phosphorus cycle and still don't know that much about the rest of it. On the plus side, though, our techniques are getting a lot better, so we can start to see things more. Measuring soil pH is important to ensure healthy crops while reducing loss and water quality problems. Testing, careful fertilizer placement are important. Again, 4 R's definitely match. Sampling depth for testing is important, and for analysis of any of the forms that I looked at. Advanced techniques like NMR and XANES have substantially increased our knowledge of the wide range of phosphorus forms, and microbial techniques are the next ones. Now, a lot of the combining metagenomic techniques with all our standard extraction techniques and NMR and XANES is just giving us a wealth of information we didn't have 10 years ago. And this, of course, will advance our understanding of phosphorus cycling. And we need to understand how changes in soil chemistry, such as pH, influence phosphorus cycling, or changes in land use or anything. We can't just measure soil test P. We need to look at the bigger picture. I need to acknowledge many, many people who have helped with these studies over the years, including my staff, including graduate students, project collaborators, staff at the NMR facility and the light source. And at that note, I will say thank you.
Thank you so much for that talk. We have had a couple questions come in the chat. So the first one is, have you looked at P losses with leaching, or is that of any concern in your area?
We don't have a lot of leaching. Of course, in the wintertime, everything's frozen solid. And in the summer, we tend to have a moisture deficit. Our soils are also relatively clay, so things don't move. You need to have a major crack or something in the landscape to get a lot of leaching.
Right, and then another question. Have producers been receptive to thinking about reducing P inputs if they have soils with high soil test P? As your work seems to indicate, they could reduce inputs without sacrificing yield.
That's the message we keep trying to say, and it depends on who you talk to, of course, and who they've talked to. There's still a lot of influence from. I still will occasionally find old school agronomists who will tell me that phosphorus is not soluble. And I can say, yeah, it's not soluble in the profile, but it's sure soluble in runoff. And there's still this need to, keep topping it up to preserve the levels, except of course what we're showing is topping it up, it's increasing it. What helps is the years when phosphate fertilizer is really expensive, because what we can show them is you could skip a year or two and it's not going to make any difference. And yes, compared to when I started with that message 10 years ago to what I'm saying now, they're becoming more receptive to it.
Yeah. Great. And then another question, is there any hope for analyzing organic P in solid state rather than extracts?
If you have a system with no iron. So yeah, it's a combination. The solid state phosphorus, it's the same problem we have with XANES, and why XANES isn't sensitive to organic phosphorus is the concentrations are too low. One of the things we can do with extracts is we extract a lot of it. We take those extracts, we freeze dry them to concentrate phosphorus, and then we dissolve them. So we start with 30 ml extract and we freeze dry it and dissolve it in 3 mL. And that is what gives us gets us past the detection limit. Solid state, you will see the main forms, which are going to be inorganic in a lot of soils, or what you won't allow you to see the smaller peaks. And you still have to get rid of the iron somehow, and that's what extraction lets us do.
And one last question. I'll also ask if anybody wants to unmute to ask a question after this one. But when you were talking about land uses, you mentioned roadside ditches. What type of vegetation were you seeing in those ditches? Was there overlap with the native grasses or anything else?
It depended. We actually have a paper published that gives a lot of detail about that. So yes, most of the time they were, well, prairies, they tended to be the prairie, where it was the tame pastures, it tended to be similar vegetation. Around the croplands, it tended to just be weeds. Yeah.
All right, thank you. Give everybody one last chance to see if anybody wants to unmute to ask any questions, but it looks like that's all that came in through the chat. All right, well, thank you for your talk today. Any additional questions coming through your chat, Matt?
Nope, no, I didn't get any other ones, any additional ones.
Okay. So thank you everyone for attending today.
Yep.
As I said, if anyone wants to contact me directly, my email address is there.
All right. Thank you so much again, Barbara, for the excellent presentation.
Thank you.